just wanted to leave some time to sort of, so there's, we can have questions and answers, hopefully. So are there any questions for any of the speakers who are still here, or for Romas, myself, or Manuel? Go ahead.
I'd like to know, which we really didn't get the answer to, how many people have been treated with gene editing technology, and where do we stand as far as, clinical trials, and, because that is real important at this point. It's even lost animals, primates, and
right. So I think you've heard, we've got one here. John, may I, at least for the stem cell part, there's just 8 in the stem cell gene therapy studies with zinc finger editing of CD34 cells. And is Jim still here? No, he's sitting over there. There is at least probably 15 or so, 20 patients that were done with CD4 editing. Yeah. The UPenn studies have been published, and Jim presented that here, at least from the published studies, yes. And I think John has not published his studies yet, but those will be forthcoming, I assume, also probably very soon, right?
There's an open trial at Case.
For HIV? Okay, any other, somebody? Okay, Matt, yeah.
So I'm going to put somebody on the spot up on the stage, or anybody who wants to, but as a result of this conference, I wonder if somebody could encapsulate just briefly where we are, where patients can take this going forward as a result of this meeting today, and sort of the, summarizing of what the results are. I mean, not, just briefly, so that, I think what I can just tell you what I see is that we're at a point where we're scaling up. We're going to need a lot more patients for trials, and so that's where I'm interested in terms of my work and my advocacy. But I wondered if somebody could encapsulate that a little bit better.
Actually, can I
please, yeah.
So Matt, I think one thing that I've been noticing, obviously, with this field, and we're obviously very, very happy to sponsor this conference, but we're not directly, Gilead's not directly involved in this space. But I am really amazed by what's going on here. But having witnessed, I've been in the HIV business for 32 years, so I basically first-handly witnessed the advent of antiretroviral therapy, combination therapy, and also the advent of an HCV cure. So what was unique about those situations is that there's an ecosystem that basically leads to this, a really fascinating ecosystem. It starts with academic researchers, government funding. Small companies are spun out of the laboratories. Venture capital comes in because they recognize there's a need. You need the funding to do that, and pharma, obviously. So that ecosystem is what drives or greatly accelerates innovation, and that's how we got to combination antiretroviral therapy and an HCV cure. So what we're missing here in this field is we don't have big pharma participation at this point, and we need that because that will really, really help. And what we really need at this point is a signal in the clinic, something that shows us that these pathways, that what we're doing here actually will make a difference and could eventually replace antiretroviral therapy. Once that happens, I think you're going to see a large number of companies come back to this space. As you know, many companies have left this space. We're down to 3 with Merck and Vivo and Gilead. There's no one else in this space. So we need these other companies to come back. The reason we need them to come back is because we need competition. We need competition because competition is really good. It really fuels people and makes things go much faster. So we're missing that part. So what I'm saying is, let's get a signal soon. If we get a signal soon, I think things can really, really take off here.
But I think there have been signals all along since the first, including the ex vivo work from Sangamo, the studies before that, the Virexis study earlier, even going back to the first Penn sort of CAR T study. there weren't cures achieved in each case, but there were clear clinical signals that were identifiable and reported. So I think that it's been green-lighting this pathway ever since. So I'm not sure what you're waiting for.
So from my perspective, those are anecdotes. Those are not well-controlled clinical trials which show you a signal, a robust signal that we can pursue. So those are good, but I don't call those signals. What I call a signal is a placebo-controlled trial with a statistically significant difference of an effective curative therapy, basically. We have not seen that yet, and that's what we need. But your point is a good one. There have been some signals, some signs that things are going on, but we need to basically have robust trials, and so that's really, really important.
But many of the other gene therapy companies, CRISPR for hemoglobinopathies or Bluebird, they didn't have those placebo-controlled studies.
I mean, we're a bit different, right?
I know, that's why I'm just...
We have combination antiretroviral therapy that works pretty well, so we need to basically rise that bar, but that bar really needs to be obviously attainable, and it needs to be safe, effective, and clearly superior to what is out there right now.
So the bar is simply higher.
Exactly, because, yeah.
Romas, actually, your comments I think are really helpful. But what I see particularly challenging about this approach, unlike some of these other pathways that you just mentioned, is it's very unlikely, I think, that we're going to find a single biotech that is able to do a phase 1 or a proof of concept and get inquired by a big pharma because the signal has worked. Because what it seems to me like is that there are so many different IP assets embedded that will come out of spin-outs, whether it's targeting, delivery, conditioning, mobilization. All of these things are kind of a one asset thing that's embedded in now one biotech company versus another. So is it RNA to be married with a targeting moiety, with what? With a carrier moiety, with another one? I mean, and to try to be a system integrator of that needs some kind of adult supervision. Otherwise, each of these biotechs are going to they're going to have their one method, and they're going to be trying to make deals with the other methods, which they don't really have enough assets and VC money to get the best piece. So composing the IP in some kind of system integrator to get it the right phase 1 or 2, that part's missing. And I think it's going to be really hard to do that with this group and the way these are spinning out, because they're all just in so little pieces right now.
I think that's a great point, and that's where the pharma piece comes in, right? You need a large company with great resources that basically is able to basically in-license all those technologies, bring them together, and that's why I was saying that's what's missing in this ecosystem.
It can't be a single Avexis acquisition, right?
Agreed.
We got a whole bunch of
Agreed.
things that need to be.
Yeah. But that's doable.
I mean, I think the path forward, as a genome editor versus a type person, some other disease is going to get T-cell editing going in vivo, or so some other condition or some other condition is going to get T-cell editing in vivo or stem cell editing in vivo, and then this field is just like ready to go, right? So that signal might come from a totally different field where the bar isn't so high to justify the trial. Does that make sense?
Are we really ready to go, though, in terms of target? I think the methodology technology is probably more advanced. We've talked about proviral excision and CCR5 targeting, but I think reasonable people could disagree on whether those are effective strategies moving forward. I'm certainly not privy to all the data, but do we need other targets also, and does that continue to be something that's holding us back?
I mean, I've heard a lot of people who are convinced, and I don't know if this is justified, but a lot of people are convinced that just enough CCR5 editing will get you somewhere meaningful. So, the stuff that we've seen in vivo with CCR5 editing, it's always very small levels of protected cells. So then you always get this skyrocketing viremia, and it's done, right? So it could just be a question of efficiency of an existing strategy. I think some of the field experts, I talk to them about this because I'm very curious. What do you do if you can edit in vivo? What do you want to do? And a lot of people seem convinced that enough CCR5 editing is going to be very meaningful. I'm not totally convinced.
So a general question about CCR5 editing, besides how effective it could be, I think we're all attracted to CCR5, number one, because both eradication cases involve perturbation of CCR5, and then there are people walking around that are homozygous for CCR5 deletions, and historically we at least thought that there was no penalty associated with that, but I'm sure many people here are aware that there was a recent study that was a meta-analysis that demonstrated that there is some survivorship disadvantage associated with being CCR5 delta 32 homozygous. And how has that affected people's thinking? I'm just curious, like, are people concerned that perturbation of CCR5 could have deleterious consequences, especially if you weren't missing CCR5 during, neonatal development or something? Curious.
So that was a very limited study, though, in a very limited group of patients from England, and they were not exposed to HIV. So it's a completely different patient population. So I think that would require a much larger study, in my opinion, to really make a definitive sort of point.
There are bars about those because the long-term consequences of triple therapy are problematic, I'm sure.
Yeah.
Well, compared to what Satish just described, it again, a larger study would need to be done to carefully assess that because, we don't know.
So, Romas, can I just take off on that a little bit? I've been harping for years about the success we had with the safety trial when we saw the doubling of T cells in the majority of people. So there are these sort of off and unexpected things that may occur in some of these trials that we would love to see taken off on by the companies. But advocates, we've worked with them and pushed them to get them to do these larger trials. And like you said, it's going to take a lot more resources, a lot more money to do those trials because they're not the original indication that they wanted from the original trial. So I just think that there's a lot of, fingers to this research that we could investigate that could lead us to the cure while we're looking for a cure. And I'm not seeing that happening because of resources and because of leadership and will from the research community.
The question that though, what about the difference between what about the differences do we know about being born without CCR5 compared to even the Moravec experience? Are they identical? I don't know.
Does anybody know? Because it's a great question, David.
Yeah.
Exactly right. You're born with CCR5 ablation and you live with it all your life versus having an intervention with a small molecule that just takes out the receptors. Actually, it doesn't really affect signaling of the receptors, blocks HIV. So,
Right.
So it's
Hi, I wanted to ask if there's any kind of situation where any co-infections that maybe make HIV easy for someone to contract give rise to maybe anything to do with the CCR5 receptor, and if so, how would someone go about identifying this before moving forward gene therapy to see if they're a proper candidate and to see how they could have better disease management control after receiving treatment to make sure you don't have a failure of treatment?
Where are the CCR5 experts in the room? This is a very good question.
And I have one more after, sorry. Yes.
Increased susceptibility, at least theoretically, when you don't have CCR5 at play. That's one example.
Also, I was going to ask, when is it better to use a CCR5 inhibitor rather than getting the actual gene edited? Especially when you're having to go through CART therapy or anything else to avoid anything that may go wrong during the trial, to find out what modality would best work and therefore guarantee patients be able to make it through that trial successfully so that the scientists could gather all the data they need.
I think that's the central question. If there was a way to ablate CCR5 and it was safe, you could basically alleviate the need for lifelong therapy, which could include a CCR5 inhibitor, for example. So I think that's the whole point. If we are successful here as this field, we can replace antiviral therapy, but it needs to be safe, and so.
Manuel?
Yeah, I wanted to ask a question. So reflecting back on the 2 sessions that we had this morning on the viral delivery vectors and non-viral delivery vectors. In the non-viral delivery vectors, we were mostly focusing on like the RNA type styles. But whatever happened to the naked DNA approaches to non-viral delivery approaches? I know that here there's like Dr. Fuller and Dr. Adair that are working on gene guns, but what about other approaches? I don't remember hearing. Or it didn't seem like there was excitement about DNA approaches to that.
Dr. Fuller or Dr. Adair want to comment?
Or like, for me, what's exciting about those like gene gun is that it didn't seem like it was that invasive of an approach as like all these other ones that we were witnessing today.
Yeah, I think with particularly gene gun, and then in general for DNA versus RNA. So RNA is just going to give you a lot more protein in a single shot. And I think when the goal is really about gene therapy, you're trying to get as much protein expressed as possible. With DNA, it's really been a lot of what's driven it is the vaccine field, because a smaller amount of protein is sufficient to trigger an immune response, and often might be actually one of the mechanisms whereby DNA are able to induce very high quality immune responses. So the gene gun actually is a way to deliver either RNA or DNA. We're exploring delivery of RNA into the skin with the gene gun. One of the problems with delivering things into the skin is that you have to get it deep enough so that it doesn't get lost as the skin sloughs off. So I think some of the targeted strategies you saw here today was trying to get things in the lymph nodes, or trying to get things that are going to go in a site anatomically that will allow it to persist for much longer. So the goal of gene therapy versus vaccines can be very different.
Go ahead.
I had a quick comment to make. I'll speak loud until the mic arrives. Whichever time I want to be into it. There we go. All right, so I think it's very exciting that innovation is happening. It truly is. I mean, CAR Ts, et cetera, et cetera. But what the field has experienced, and I'm saying this from somebody who has worked at Juno Therapeutics for 4.5 years, is manufacturing advances very slowly. And there's a reason for that. The contract manufacturing organizations are supplying customers, and Autolys's, for example, CAR T-cell therapy program got delayed by 5 months in April, resulted in a 20% stock crash. Forget about that. The patients, et cetera, are going to have to wait. So I wonder if there's a way, perhaps Hans-Peter and Keith, perhaps in one of the next cycles, to try to bring some of those manufacturing organizations here as well. Aldevron, the GMP plasmid supplier, has just opened some new ground to really advance their innovation, which is really encouraging. So, so far manufacturing has been chasing the field, and it really has to catch up, because I don't think innovation is going to be a bottleneck. It's really going to be that supply. I wanted to add that comment, and Romas, I mean, you've been in this field. I'd love to hear your thoughts. Maybe for small drugs, this wasn't really a problem, but for these complex therapies, it absolutely, and you too, Leah, from Regeneron, right? So thank you.
Yeah, no, thank you very much for bringing that forward. That's a great point. You're absolutely right. For small molecules, and we've done many of those, that part is often the easiest part, right, the back end. Once you've got the molecule, it looks great, the formulations, et cetera, is relatively simple. For this field, you're absolutely right. This is much, much more challenging. I was thinking of antibodies as well, somewhat more challenging than small molecules, but nonetheless easier than what you're describing. But you're right, when you basically start to manipulate cells, and it's a whole different ballgame. So yeah, that'll be another hurdle, but hopefully, again, if we have a signal that people believe in a well-controlled study, and people say, okay, we've got it, let's go, great people come forward, innovate and solve amazing problems, and we've all seen this, but there has to be a real drive to do that, and for that there needs to be obviously an impetus, and that would be a signal we believe in.
Okay, maybe 1 or 2 more questions. So I want to get back actually to Matt. I'm not sure that we answered your question. I did hear it though, very clearly. So I would go back there and say we're moving back into clinical trials. Monique presented beautiful data, and what we know from the transplant studies, we know CCR5 and donor immunity can cure HIV. Now we can do all this again with the patient's own cells, ex vivo or in vivo. We know where to go. Make the cells, CD4 cells resistant. We can make CAR T cells or therapeutic vaccines. I think we know where to go again. And I think, we know there's already some CAR T cell studies in the clinic again. I think we'll see this coming back into the clinic. Ex vivo first, of course, but then hopefully soon also in vivo.
Thank you.
Is that what?
So pretty much everything we've heard about involves patients who are suppressed on ART, and we're giving them some therapy, or potentially giving them some therapy, they're doing an analytical treatment interruption or so forth. But there are a lot of people around the world who are untreated, so maybe get some opinions around the room about using a curative therapy as a first-line treatment and potential pitfalls or benefits.
Matt, do you have any thoughts?
That's not my, I don't think that way because I'm such a, far into therapy, but that's a great idea.
Yeah.
So if I may, obviously we need a curative therapy. We need it to be safe, we need it to be effective, we need it to be relatively cheap. We don't have that right now. So right now the issue is how do we get antiretroviral drugs to those who need them. That's a daunting problem, but it is still the simpler of the two problems because we have no solutions in terms of cure, none. So when we get that, then we can revisit that, but we have to make sure that that it can be basically utilized in developing nations, but we don't have it right now. So
maybe one last, yeah.
I kind of want to end where we began with Dr. Johnston's presentation on kind of the closed networks, on kind of where publication is and where study is and where the science is, and on who's studying around genetics and cell and gene therapy and on cure. It's a very limited group of scientists that are doing this work, and I think it kind of goes to the question you just posed to Matt. And I think if we're going to figure this out, we have to think about it in terms of access and engagement, both for the scientific community as well as community engagement, and think about it in a very strategic way. How are we getting new communities involved and thinking about this, new scientists involved and new communities involved? And I think we have to break those networks and get new scientists involved and get new communities involved and get them access to this science. So I think that that's a key piece of this as well. I think it's not just getting new companies to be there, it's to get new communities there, it's to get new scientists there, and to get new biotech firms there as we're trying to figure this out. Because it's not just the CCR5 theory that's going to get us to a cure. That's where I wanted to leave us.
Thank you very much. And I think with that, I'd like to conclude our meeting. We'd like to thank for today, again, very much the Bill and Melinda Gates Foundation that sponsored today. And I know Keith will mention all the other sponsors again for the overall meeting. One last thing I wanted to say is also I've been really impressed with the attendance. It's almost as full as when we started today. This has been truly amazing for me. Thank you very much.
Yeah, so in addition to thanking the Bill and Melinda Gates Foundation, we also have our platinum sponsors, so we're grateful to Gilead, of course, Abbott and Amphar. Thank you very much. Gold sponsor was Marriott, and Bayer, who was our silver sponsor. I want to take just a sec and congratulate our poster winners for the 3 top posters. If you're here, stand up. Let's see, the first was Mary Pumpash from the University of Minnesota. Congratulations. Colby Maldini from the University of Pennsylvania. All right. And finally, Tonya Potit and Karine Dubé from Hopkins and UNC. Well done. All right. Thank you, everybody, for joining us. We've heard a lot of great science. We'd love your feedback. We'd love to get you back out here. If it seems like this is something you want to do again, then we would love to host you. So thank you for coming to Seattle, and have a safe trip back. Bye.